Hey everyone, welcome to my molecular biochemistry series, and in this episode we are focused on DNA mutations. In previous episodes I talked about the molecular structure of DNA. I touched on how DNA will replicate naturally within the cell. We even went over a laboratory technique called PCR, or polymerase chain reaction, which will amplify DNA, make many more double helices, so that we can more easily analyze them or even manipulate them for our purposes. But in this case, we are thinking about mutations, and the first mutation we will discuss is called a silent mutation. And it is aptly called that because we are modifying what is called the wobble position, which is the third base pair in a sequence of base pairs, that even if it's mutated or changed or altered, it will still code for the same amino acid. And if you have the same amino acid being outputted, there's no structural change to the protein that is being translated. And so there's no issues that will arise with this sort of mutation, and so it is aptly called a silent mutation. The next mutation we will think about is called a nonsense mutation, and I use the phrase stopped nonsense to more easily remember what this mutation does. And what it does is prematurely code for a stop codon. And a stop codon is something that tells the ribosome to stop linking and creating amino acids. And so if you do this prematurely, you're going to get a protein that doesn't have all of its necessary structural components, and it will misfold and become non-functional potentially. And if you have many non-functional proteins, the cell itself will become non-functional, and then it won't be able to do what it needs to, and it can cause disease and disorder. In this scenario, we altered the first base pair in a sequence of 3 to an A, and so the mRNA that's going to come in through here is going to be AUC. It's going to move from the 5' end to the 3' end of the template strand, and it's going to mimic the coding strand, except anywhere there's a T, it will become a U. And so this is transcription, and then this mRNA will undergo a little bit of modification and then be the fully complete mature mRNA and enter the ribosome, where it then will be translated. The next mutation we will briefly touch on is the missense mutation. And again, this is a point mutation where we are modifying a single nucleotide. In this case, we are modifying the second base pair in a sequence of 3 to a G, and this will code for a different amino acid than what was previously being coded for. And so specifically, I think in this scenario I made it to be a glutamine to a valine, and this is commonly seen in sickle cell disease. So a missense mutation can be very problematic. The next and final mutation we will discuss is a frame shift mutation, and it is when the cell inadvertently either adds in one or more nucleotides or takes away one or more nucleotides, and this will shift the reading frame of the DNA such that when the mRNA comes through, it's going to code for a bunch of different amino acids in the wrong sequence. It's shifting how it's being read, and this can be very problematic, and we see it in cystic fibrosis, Tay-Sachs disease, and many others. And so a frame shift mutation can be extremely problematic, and so this is something to keep an eye on as we think about DNA repair mechanisms, which I will talk about next. So that is a great overview of DNA mutations, and to quiz yourself on what you just learned, what were the 4 common mutations, how do they work, what is their process, and see if you can recall that, so then later on down the road, days from now, weeks from now, you'll be more likely to remember it. And with that, I'll see you guys in the next one. Bye.